All right. Well, it's 12 o'clock, 12 noon in Los Angeles, and welcome to the end of the year roundup for Enigma. We really appreciate folks joining in a very busy week from all quarters of the world, and we'll do our best to fit this into an hour. It's going to be a review of all the wonderful things that have happened this year across the world as part of the Enigma Consortium. And first, a big thank you to the folks that you see listed here. These are the local Enigma team here in Los Angeles. Um, as there are a few new folks, so there are people joining who maybe have not been with Enigma for a long time, I'll give a little bit of a recap on the overall landscape of Enigma. We have 2,000 or so participating members from 45 countries, analyzing over 100,000 data sets. And the folks are organized into 55 or so working groups, some with a clinical focus, some looking at aging and development, adult and adolescent psychiatry, some focusing on genetics and genomics, and 14 groups dedicated to algorithm development. And you'll see organized here the sort of layout of the different groups. Some have subgroups. So the brain injury working group, as you see there in the middle, has a number of different focus areas. So does anxiety, and so do a number of the others. And up at the top, you'll see the groups that develop and test algorithms for analysis of all the data sets that you see below. So here's where the data is coming from. Someone asked if we have a site in the Canary Islands in the Atlantic Ocean there, which we should definitely all meet at next, and apparently we do. So thank you for joining from the Canary Islands. We'll begin with a little bit of a roundup of new working groups. So these two pictures, as you've seen here, were in Montreal at OHBM earlier in the year, and also at SOBP, the Society of Biological Psychiatry in San Diego in April of this year. And I'll give you a little bit of introduction of the new groups this year. So Enigma Language is a new effort set up by Simon Fisher, Mike Ullman, Christos Pliatsakis, and others who you see listed here, to examine the neurobiology of language, to investigate links between brain structure, genotype, and language measures. They'll be looking at whether brain structure and genetics predict language performance, whether being bilingual or multilingual, as many of you are, influences your brain or health throughout life. If you're interested, do fill out a form or contact them. A kickoff call is planned for January 2024. And there's a survey. So it may be that the cohorts that you are collecting data from, or your colleagues are, have collected information on the number of languages a participant speaks or understands, how old they were when they first exposed to learning the language, and how fluent they are. So that'll be a very exciting project. Another new working group led by Tony King and Sabra Skarsan is focusing on meditation, contemplative practices, meditation, yoga, mindfulness. And they're putting together a position paper about their plans to analyze the brain correlates of contemplation, a very, very exciting new project. Some of the working group chairs have pioneered work mapping the functional topography of meditation, and they're very interested in understanding the therapeutic use of meditation and contemplation. And it's a very fun working group. You can see some of the members of that group here. Nicole Peterson and colleagues listed here and shown here have set up a group on neuroendocrinology to examine the relation between puberty, sex hormones, and contraceptives on brain measures from structural MRI, T1, T2, diffusion, and resting state. There are several areas of focus. I'm looking at puberty, menstrual phase, pregnancy, maternity, and menopause. And if you're interested in joining or have data, please do take a look and fill out their survey. And here's several members of the group at their kickoff call. A new group led by Valentina Lorenzetti from the Australian Catholic University is investigating experimental cannabinoids, either experimentally administered or administered in different ways, and their link to brain integrity and behavior. Obviously, there are both medical and non-medical uses of cannabinoids, and we're looking forward to a lot of interesting work from this group. Jan Kide and Sylvia Gustin, along with Nada Jahanshad, have started a group on chronic pain. They, with their colleagues, are looking at different types of pain: migraine, back pain, spinal cord injury, neuropathic pain, and looking at brain correlates in over 30 data sets from across the world. They also recently submitted a grant to support this, and if you're interested, do fill out their data query. ENIGMA Gradients is a new group focusing on the analysis of brain connectivity. Now, if this is new to you, do check out work by Sophie Volt, Matthias Kirchner, Clara Moreau, and Boris Bernhardt. It's a fascinating area where the functional connectivity of the brain, which can be mapped using functional MRI, can be analyzed in such a way that the circuits and systems, many of you are familiar with the default mode network, different resting state networks, they can be laid out in a topographic way. And if you have any spare time over the holiday, you can read some of the work by Dan Margulies, who was one of the first proponents of this. It allows you to visualize the functional geography of the brain in a way that is very simple, rather than the activation foci being distributed all over the place in the brain. You can see the layout of the brain's networks. And a lot of the evidence is that in some disorders, there's a contraction of the so-called principal gradient, which means that the functional synchrony moves elsewhere in this circuitry. So we're very interested in working with Sophie Volt, Matthias Kirchner, and Clara Moreau to provide tools to integrate the analysis of gradients and principles to understand both disorder-specific and cross-disorder differences and how they change with the evolution of disorders. So this was a very fast first abstract by Sophie Volk and Matthias Kirchner with the addiction working group, which is led by Hugh Garavan, looking at, first of all, for different types of substances of abuse that you see there, alcohol, amphetamines, cannabis, cocaine, nicotine, and opiates. There are thickness differences in the gray matter of the brain, but they're also similar. And not only are they similar, they resemble certain hubs or covariance structures of the brain. And there's been a great deal of interest in understanding whether or not these coordinated alterations link to the patterns of connectivity that you can then examine using their toolkits. So moving on to grants, there are 30 grants now supporting ENIGMA, some from the National Institutes of Health in the United States, the IMH, Neurology and Stroke, NIA, National Institute for Environmental Health Sciences. You'll see also listed here, there's grants from the German government, the Deutsche Forschungsgemeinschaft, the Alzheimer's Association, the Australian NHMRC. And thanks to all of you for spearheading this. You'll see a vast array of neurological, psychiatric, and developmental grants that are listed here as active. 6 grants were awarded this last year, and in 2022, 6 grants were awarded, some of them focusing on the environment in the brain, bipolar disorder, Tourette syndrome. A couple of grants to Raj Mori and Mark Logan, their team, to look at post-traumatic stress disorder. And a number of new grants that focus on copy number variants in the genome, the relationships of genomics to white matter, and predicting therapeutic response in depression in severely depressed patients treated with TMS. So these grants look like they'll be awarded. Congrats to the folks leading the schizophrenia and schizotypia and addiction and Parkinson's working group. Dr. Hugh Garavan, Patricia Conrad, and Scott Mackey heard news that they just got a very good score of 12 on a scale of 10 to 90 for their ENIGMA addiction renewal. There's also a new grant to Jess Turner, Theo Vanna, Gemma Modinos, Andrei Alleman, Matthias Kirchner, and Andrew, not Zaleski, who have been awarded a 5-year grant to study schizophrenia and schizotypy. And new grants that scored very well looking at dementia and Parkinson's and eating disorders to the folks that are listed there. We're awaiting word on reviews of an emotions grant in OCD, a sex differences grant in cross-disorder developmental conditions. The Milken Foundation has been asked for funding to look into lithium treatment response. And also a very interesting initiative in Pakistan led by Maheen Adamson and Mohammed Parvaz with the Aga Khan University and a team in Peshawar in Pakistan to scan people there. We'll look into that a little bit later. And some supplements for the World Aging Center to support Emily Dennis and Lisa Wiles' ENIGMA U Global Education Project and an AI project in bipolar. NIH will get a lot of grants early next year. I won't list them all, but just for your records, a long list of grants that are going back in February or March and the various scores that they received over the last year. 26 new journal papers in the last year. Congrats to all of you. When you receive these slides, and Sophie will send you a PDF, the link should work, so you can browse through work by your groups and your colleagues. A very, very impressive range of work that is listed here in these 26 papers. 3 just accepted, one on deep learning and major depression, identifying features in neuroimaging that are associated with major depression by Vladimir Belov and Roberto Goya Maldonado here. Also work by Rebecca Carestes and Ian Harding and Enigma Epilepsy, examining cerebellar atrophy in a sort of mosaic across the cerebellum. And also work by Ashley Huggins and Raj Mori and colleagues examining the cerebellum in PTSD. And we'll look at their results in a minute. And then 6 preprints that have also been submitted. We'll cover a handful of these. Some of these projects are pre-registered, so I wanted to sort of bring this up. Enigma's secondary proposals are a form of pre-registration. You say exactly how you're going to analyze the data. But several groups, the anxiety group, the sleep group, the antisocial behavior and conduct disorder working group, have been pre-registering studies, which means you can essentially say what you're going to do, and if you expect small effects or have a non-obvious hypothesis, you'll avoid allegations of fishing for results until you find something significant. And the anxiety group, led by Jana Marie Bass Hogendam, was praised in a journal for doing this. There is some extra time involved, but it'll certainly help if you want to have some guidance in how to do it. So we're going to recap some of the methods being used today in Enigma to see if they're of interest to new people who might want to try them. There is a basic set of methods for structural MRI, diffusion MRI, and functional MRI analysis that you see organized here, both voxel-based and surface-based and regional analyses, more recently supplemented by workflows for EEG and MEG. And we'll talk a little bit about some of these newer methods. People are trying out methods to do normative modeling, look at gyrification. There's a new genome-wide association study of gyrification just about to begin. Some deep learning methods developed by Neda Jahanshah's lab to segment and partition the corpus callosum, and a very large array of studies looking at structural covariation of the brain in the groups listed there. The repository for these methods has been updated. Thanks to all of you who've populated this. If you have a protocol that's being used, please let us know and we'll house it here. And do click on the link. It's been very nicely reorganized, and you'll see a very easy to navigate set of scripts available for the different modalities there. Big thank you to Leah Waller, Henrik Walter, Ilya Veer, and Suzanne Erck at Charité in Berlin for creating a very widely used workflow for the analysis of functional MRI. Both resting state and task-based fMRI can be analyzed using half pipe. There are manuals. If you look on the right, if you're doing task-based, resting state fMRI, or even just quality control, there are guidebooks as to how to process the data through many stages. Big thank you to Leah and Ilya for spearheading this. And very easy to use scripts and workflows. And there is a mailing list for updates. If you have people analyzing functional MRI in your group, do encourage them to subscribe to the mailing list shown here, and they'll be getting updates and advice on how to run workflows. Here's a dashboard of how functional MRI data is being analyzed right now across the many clinical working groups in Enigma. As you can see, a couple of manuscripts have been published or being revised. Congrats to Willem Bruin and Guido van Wingen in the OCD working group for being the first to publish a multi-site analysis. And you'll see a number of the groups across psychiatry and neurology and development have been beginning their work, even one in infants using infant-tailored scripts. Now, this leads to an opportunity. If a lot of the clinical groups process functional MRI in a similar way, results can be compared across clinical conditions. Lifespan charts can be made to describe the development and aging of the brain in terms of its connectivity. So we're wanting to have quarterly updates to review some of the findings of the functional MRI analyses with a view to harmonizing some of the atlases, seed settings, some of the ways that the quality control is handled, which I think will essentially lead to a lot of opportunities for cross-disorder work. So look out for an email about joining some of these meetings where you'll hear about colleagues analyzing functional MRI. There is a recommendations paper, Karen Kayenbergs and Frank Hillary, as part of the traumatic brain injury working group, have set out 7 tips to make sure your findings are reproducible. That's wonderful. Do have a look at that paper. As I said, congrats to Willem Bruin and Guido van Wingen for their functional imaging analysis of functional connectivity in obsessive compulsive disorder. They found hypoconnectivity or abnormally low connectivity, primarily in sensory motor networks with a fairly robust effect size, and a few hyperconnectivity, overconnectivity, mainly to the thalamus, which is very strongly implicated as a hub in OCD. So it's very interesting to see these results coming out. Switching gears to the cerebellum, 3, actually now 4 papers have been analyzing the mosaic of regions in the cerebellum and how they're affected in a variety of conditions here, brain injury, Parkinson's disease, and epilepsy, just accepted. So congrats to the protagonists of those papers. Just to show you what the results look like, this study of traumatic brain injury by Finian Kelleher and Emily Dennis and their group essentially shows that in mild TBI versus moderate and severe TBI, the cerebellar changes recover. And so if you look at the acute and chronic stages, you'll see indicated in red is the level of atrophy, and look at the tendency for the groups to recover if they're more mildly affected. In Parkinson's, there's another fascinating result that early in Parkinson's, in stage 1, there's a hypertrophy. Now, these regions of the cerebellum are larger than in typical elderly people of the same age. But this is then overtaken by the progressive degeneration that is famous in Parkinson's. So we're very interested in whether the hypertrophy early on is due to inflammation or compensation, or even an early genetic protective effect of Parkinson's disease genetic risk. Ian Harding and his team have been studying ataxias, Friedreich ataxia up there on the top left, with cerebellar deficits that depend on the duration that the patient's been ill, and also the age of onset, earlier ages of onset being more severe. And you can see they're also compiled a library, so to speak, of all the spinocerebellar ataxias. And the little colored bars there on the bottom indicate sectors of voxel-based cerebellar maps compiled using the SUIT cerebellar mapping toolkit. Also in bipolar disorder, Charles Lady and Jocelyn Huenu at Neurospin in Paris have worked with Chris Ching, Ole Andreasen, and the bipolar working group to map the mosaic. It looks like maybe lobules 7 and 8 are being affected there. With one interesting qualification, that the people that are very depressed in bipolar disorder have cerebellar deficits, at least structurally, but those with bipolar did not show a case control difference. Very, very interesting finding. And here's a sort of inventory of where people are at with cerebellar analysis, much thanks to Ian Harding and Rebecca Carestes for creating these scripts that so many groups have benefited from. And you can see some colleagues to reach out to if you're planning this type of analysis. There is a spinal cord toolbox. So this is very interesting. It's led by Tiago Rezende and the ataxia group. You may wonder if the cervical vertebrae are present in your MRI scans, but they very commonly are, at least C1 and C2, as you see in some of the images here. And these have led to some of the most robust brain clinical correlations across all of ENIGMA. You can see very well-behaved correlations between clinical disease severity, at least in the ataxias, and morphometry of the spinal vertebrae. So do have a go. It's being tried in Parkinson's. If you have a disorder that might affect the spinal cord, do have a look at the spinal cord toolbox. So this has really been a good year for diffusion MRI. So this is Alyssa Zhu from Neda Jahanshad's lab here at USC explaining DTI harmonization at the SOBP ENIGMA workshop in San Diego. There are 11 papers now published. If we missed any, do send us a reminder. The green ones have been published. Do follow the links to have a look at what has been found regarding brain microstructure using diffusion imaging. Just as a flavor of what is being found this year. So this is a monumental analysis by Connor Owens-Walton and colleagues in ENIGMA Parkinson's, where diffusion MRI data from 17 cohorts was analyzed. And you can have a look at a very curious effect that goes on. So the fractional isotropy, a measure that's normally low in neurological illness, is actually higher in the first stage of Parkinson's disease. And then it's progressively overtaken by the degenerative pattern that is more familiar in the dementias. And so again, very curious and interesting finding in early PD, which has now been replicated here with DTI in the cerebellum and also in the morphometric work that was published earlier on. Talia Neer and Neda Jahanshad and their colleagues have been studying HIV, looking at the white matter microstructure, integrating data from across the world. This is a new analysis that they just finished, that they found. This will be no surprise to HIV researchers. They found that microstructural deficits were linked with CD4 T cell counts, but not with viral load, because most patients are treated with combined antiretroviral therapy, which suppresses their viral load, but their NADA CD4 T cell count is usually linked with the degree of brain deficits. And also there's some evidence that these deficits interact with aging, which is interesting for therapeutic management of people living with HIV. Some new toolkits on the horizon. So we're about to launch early next year an ENIGMA tractometry toolkit. Gaon Kim and Kathy Lawrence have tried it here in autism spectrum disorder, mapping fraction isotropy deficits across the left arcuate fasciculus, a famous language pathway. And also they've been working in ADHD, showing the profile of microstructural differences, mainly deficits across the white matter. This has been contributed to from many quarters. So Bram Chandio, in her PhD work with Eleftherios Garifelidis, who runs the DIPI toolkit, and also Iyad Bargari, who works with Neda Jahanshad, and Wendy Feng in our lab, have all been writing tools that would not only map tracts in your patients, but also integrate multi-cohort tract data in such a way that the methods harmonize the data along the tracts. I mean, you can imagine a technique such as combat, which many of us use for adjusting for site effects in multi-site data, that could be generalized to tracts, and there'll be soon methods available to have a look at that. So some beta testing is being done now. Here's nice work by Leila Nabulsi, looking at bipolar disorder and its effect, with gratifyingly increasing effects as you add cohorts. I mean, if the effects went away with more data, that would be very bad. But she's finding in these QQ plots here that the effects here in the frontal cortex, which is hypothesized in bipolar disorder to be in deficit, are increasing. So this work also is with Professor Frank Ye, who many of you know as the author of DSI Studio. Neda Jahanshad and colleagues have recently won an R01 to support the application of these methods and others across various ENIGMA working groups. Kudos to Sophia Franco and Riyang Yi and their team for publishing Centile Brain. It's in press at Lancet Digital Health. It integrates data from many tens of thousands of controls from cohorts across ENIGMA to develop normative models. You can use it to norm your data. This is related work just out by Julio Villalon and Alyssa Zhu, where they've been integrating over 50,000 people's white matter microstructure measures, harmonizing it and giving out tools where people can use these templates to normalize data from clinical cohorts. The results look a little bit like this. So, I mean, as you can imagine, white matter microstructure has much the same growth plateau and degeneration as many of the other measures. But in C here, you'll see how different sites have different trajectories. So depending on the protocol that they used, which might include large or small voxels or different numbers of gradients, the mean and variance of the derived measures from each protocol can differ. And so that's exactly what these tools are designed to address. So here's some nice work by Julio Villalon, where there's some algorithms that essentially take the data over there on the left, which are the diffusion measures from different cohorts, often very disparate, and compile them not just into an average trajectory, but one that fits data from each site and generates z-scores or z-scores for abnormality quantification. Two toolkits for brain-wide mapping of structural anomalies: the ENIGMA VBM tool, developed by Matt Kempton and Anthony James, and the CAT12 toolbox that's been developed by Christian Gasser. Many groups across ENIGMA shown here have had wonderful results with these, obviously taking to a more brain-wide level some of the findings that have been mapped before. Just to interest you in trying this, so Matt Kempton and Shane Prakar ran VBM in bipolar disorder. And look at that tremendous result in the posterior corpus callosum. I mean, of course, there are frontal deficits in the gray matter in bipolar disorder, which is quite expected. But the intense focus of deficit in the posterior white matter in the corpus callosum is very interesting. This is a composite, a meta-analysis of data from 25 cohorts. So do reach out to them if you want to try this in your working group. It's been giving very, very good results. Christian Gasser also, his CAT12 toolbox has been providing surface-based analyses and many, many other types of analysis that you can read about in this preprint that are described here. This is over 20 years of work, and a lot of people in our lab really like using this toolkit, so it's highly recommended. Some of the analyses of brain structure have begun to probe more deeply the nuclear substructure of the thalamus, the amygdala, and the hippocampus. And you'll see a roadmap of how to do this for the hippocampus by Philip Sayman and colleagues down there on the bottom left. And also very promising results in stroke, OCD, bipolar disorder, and addiction, showing that the sectors of these structures can often have differential effects that are worth inquiring into. So this is an example from Vilde Brecker and the OCD team, where the amygdala and the hippocampus are partitioned. And in some cases, the overall volume is the winner. It shows the greatest effect. But in others, like the amygdala on the left, there's a subnucleus that shows stronger effects than the entirety of the amygdala. So there's much promise in applying these toolboxes, which have been organized into Docker containers by the OCD group for all of you to use. Brain age is the topic of a working group led by James Cole, who is one of the originators of the method. Across ENIGMA, there are methods that compute brain age from regional ROI measures there at the top. You can even use a website called Photon to compute that without sharing data centrally. Brain age R is a new method that James Cole and his group have developed that uses the raw T1 weighted MRI. And many groups are trying a variety of these to derive estimates of the brain age, which is the apparent age of the brain based on its structure, and shown that in many disorders here in major depression, apparent brain age appears to be shifted towards older ages. And you can upload your data into many of these tools. Here's the Photon toolkit. And here's Laura Hahn presenting her work that shows an effective aging of over a year in the brains of people with major depression compared to matched controls. And this is a really nice set of tools. Here's Sukli Liu, who chairs ENIGMA Stroke, found that a younger brain age was associated with better outcomes after stroke. And I mean, this is a paper that's just out in Neurology. You can read about this across 38 cohorts, not just sensory motor deficits, but all domains of function were linked with earlier brain age. Very interesting. And there are the authors, rather a large number of people who contributed to this ENIGMA stroke study. Much credit goes to Brenda Bartnick-Olsen and Alexander Lin for creating a spectroscopy workflow for ENIGMA, which they review in this overview paper here. They're recommending the Osprey pipeline for single voxel data, and they're finalizing a number of scripts that any of your group members that have MRS can use. They're very interested in groups with even single voxel data, any age, any disease, any region. Do contact Brenda and Alex. And they're all going to start to do a lifespan study of controls. So just to kick off this group, to look at the trajectory of molecular measures from the brain with MR spectroscopy throughout life. And thanks to them for creating a very nice set of tools to do that. Anyone should contact them if they have MRS data and interest in these projects. Alison Nugent, Jeff Stout, and Anna Namist at the NIH have developed a fabulous toolkit for MEG, magnetoencephalography. If you have data, they're very, very interested in running these workflows on your data or showing you how to do it. And this is going to be the beginning of really a wide range of both normative and clinical studies using MEG. Taylor Kuhn and colleagues have started a new group on neuromodulation, deep brain stimulation, TMS, and a number of methods that you see there on the left, to try and understand the neural systems that are affected by interventional neuromodulation and their emotional and behavioral effects. They're beginning to analyze longitudinal MRI data where neurostimulation has been tried. And they're also very interested in optimizing stimulation parameters. Obviously, if groups have been doing this with different methods, a meta-analysis could be used to evaluate whether or not the stimulus parameters influence the results, and if so, what would be the best thing to do in the future. And so they're planning a grant. Nicole Peterson and Taylor Kuhn at UCLA, do contact them if you want to be involved. And an invited manuscript also, which is now collaboratively being written. Now, distinct from neuromodulation is neurofeedback. So David Mailer, Emily Haug, and David Linden have been collating data sets from across the world to discover what regions of the brain are involved in self-regulation during neurofeedback training. It's a very interesting set of questions. They also are interested in MDD, substance use, ADHD, PTSD, and schizophrenia, for which there are a fair number of neurofeedback data available across the world. So do sign up if you want to. There's a query form for you there. I want to tell you a little bit about new ways to harmonize data from multiple sites, not just images, but also clinical scores and other metrics. So these two papers out in the last year are quite useful. Elizabeth Haddad, working with Neda Jahanshad at USC, tested different versions of FreeSurfer and verified that they're all good. And if people are processing data with different versions, you can have a look at that and be reassured that the metrics are not only accurate, but can be combined in the same study. Delin Sun has been comparing the Combat family of tools to show that Combat GAM, which is the nonlinear version of Combat, should be used, or at least gives better results if you're studying an age range, at least where the effects on your phenotypes are nonlinear. That's a nice reference to justify using Combat in your studies. And this is a wonderful guided tour of site correction methods or harmonization methods for imaging, authored by Johanna Bayer and Philip Sayman, where they've reviewed all of the major methods to integrate data across multiple sites, adjusting for site effects, nonlinear age effects. Some of them, as you can see here, use deep learning, generative adversarial networks, a lot of very complicated methods. And you'll see their strengths and weaknesses reviewed, and plus a number of places that you can download these methods if you want to try them. Another sphere of activity is harmonizing clinical data. And so if you are assessing a disorder, it often is assessed using multiple scales. Some sites may collect data using one clinical assessment, whereas other sites may not use that. And you may be in a position where you want to impute or estimate scores that would have been obtained if different surveys had been used. So Emily Dennis, Eamon Kennedy, and their colleagues in the TBI working group have been developing calculators, methods, statistical approaches to do this. Do have a look at those. One I wanted to tell you all about, which I think is just thoroughly remarkable, is based on the transformer architecture used in ChatGPT. So supposing you had a questionnaire that evaluated your symptoms, and someone else was evaluating people with a different questionnaire that evaluates basically the same symptoms. Large language models can estimate the semantic similarity between questions. And I mean, we know they can do this. They can summarize, they can even answer questions for you, but they can summarize text, and they know which sentences are similar. So what Eamon Kennedy and team did, if you look on the bottom left there, is they took an inventory of questions on a clinical questionnaire and paired them up with another inventory, which is designed to assess broadly the same symptoms, and measured the syntactic similarity. And so if the question was evaluating basically the same thing, it would give a score to the other questions. And then it would predict scores that were not collected based on knowledge of what the similar questions were in the data set that were collected. And so this is truly remarkable, a very, very clever way of doing it. And I actually grew quite excited because many of the groups that collect clinical data from other disorders could pour their questions in and see how it does. I mean, it was just a fascinating question. So kudos to Emily and Eamon and their colleagues for coming up with this method, which to me is quite novel and remarkable. Colin MacDonald, Dara Kennan, and Sinead King have been harmonizing medication data. Now, if you look at the top right, these are the differences in subcortical volumes in people with bipolar disorder, showing that in bipolar disorder, you have a smaller hippocampus. But do you? So if you look down on the bottom right, Once you've stratified patients into people taking no medication or lithium or anticonvulsants, or for that matter, antidepressants or antipsychotics, you'll see a very different picture. So, in an attempt to get into this, Chris Ching, Colm MacDonald, Joaquim Radoa from Barcelona and colleagues have been leading the ENIGMA Bipolar Medications Initiative. And if you're dealing with medication issues in the disorder you study, do reach out to them. They've used the neuroscience-based nomenclature, which is a recommended system for categorizing medication. And you can see that faced with a intractable array of different medicines that might be prescribed often at the same time, it gives you a sensible set of mappings to how these medications act in the brain. And then the premise is that these might link with greater signal to noise to the brain measures that we're examining. So do reach out to them if you're interested. Moving on to genetics, there are a lot of GWAS studies. So this is a remarkable study of almost 75,000 people under revision at Nature Genetics that has discovered over 500 single nucleotide polymorphisms in the genome that are associated with the volumes of various key structures in the brain. Kudos to Luis and Adrian and their colleagues for doing this. They also are finding tremendous overlap between risk genes for Parkinson's disease and genetic loci that affect the brain. Also links to ADHD, insomnia, and neuroticism. Very, very, very interesting paper here. And not only that, you can predict the volume of different structures of the brain using a polygenic score that's derived from these GWAS. So just to summarize, they performed an analysis of genomic loci that are associated with regional brain volume. They added those scores up, at least for genes that make a difference. And then they can explain up to 10% of the variance in many of these brain metrics, which is truly remarkable. Now, there's some caveats. It doesn't work in everyone, and they're trying to beef up the numbers of non-European ancestry people in their study. But do have a look at that. It's a very, very fascinating paper. A white matter GWAS by the folks shown at the top is just about to begin. If you'd like to do GWAS with DTI, do fill out the survey. A data freeze has now happened for Ian Harding and Sarah Medlin's team's genomic study of the cerebellum in 60,000 people. They're also interested in rare variants. So if you want copy number variants called in your data, do contact them and they'll link some of those rare variants. The spinocerebellar ataxias in particular are of interest. So a huge congratulations to Vilta Beltramaitita and her colleagues for doing a successful genome-wide scan of brain age. So this used Laura Hand's ENIGMA brain age model and found loci in the genome that are reproducibly linked with brain age. A very, very interesting study that they're just wrapping up. Also, Philippe Jawinski, Dirk Smit, and colleagues have been running genome-wide association studies of a variety of EEG measures, different spectral band frequencies that are affected in psychiatric and neurological diseases. And they've discovered two genome-wide significant loci, including one that was already implicated in schizophrenia. And I know Sanjay Sisodi will be very interested in this. He's one of the chairs of the ENIGMA epilepsy group and is interested in the genetics of epilepsy. They found that some of the loci implicated in generalized epilepsy come up as having a direct influence on measurable electrical activity on EEG. And this is in healthy people, so it's very interesting. You'll see that row of blue circles shows that all the links or all of the major links between EEG genetics and disease genetics come up in epilepsy, which is very, very interesting and logical connection. They're also looking at the links between EEG genetics and other brain metrics, showing that it's actually quite tight with morphometry, a little less so with functional MRI, and that there's cross-heritability among the measures. And they're beginning to actually create normative charts from these measures that should be very useful. Leah Waller, Henrik Walter, Suzanne Erk, and Ilya Veer have been doing genome-wide association studies of task-based fMRI, and they just processed a phenomenal amount of task-based fMRI. Now, if you look at these tasks, the tasks involving emotional faces and working memory, even the brain mapping results without genetics are fascinating. I mean, a study of this magnitude with this number of subjects is revealing the the activation topography of these tasks with a rigor not previously seen, and then with a sufficient number of people that you can do genetics. Now, one of the challenges is to come up with an analysis that doesn't endlessly proliferate. So I think Leah and her team are trying to process data in such a way that they've covered all of the bases. You know, it includes data from European, non-European people, and looks at the data in a number of different ways, but also, you know, to prune down the number of analyses that they'll end up wanting to do. Sylvain de Rivière and her team at King's College London have a remarkable finding. And so they have been linking genome-wide epigenetics to structural volumes of the brain, including hippocampal volume. And they discovered that the methylation of a number of homeobox genes predicts regional brain volumes. Now, if you did developmental neuroscience as a student, you will have been told about homeobox genes and segmentation during embryogenesis. They have actually found that they look genome-wide. They have genome-wide hits for regional brain structure. These plots are a little different than GWAS. So the locations in the genome where methylation is different and is associated with the volumes of different parts of the brain. And you can even map methylation status right onto the brain here, as you see in the cerebellum. So congrats, Sylvain, to you and your team for these truly groundbreaking studies. Very, very interesting. Ida Sonderby and Ulla Andreasen have been working on copy number variants. So these are deletions or duplications in the genome that usually are quite rare. This truly remarkable study by Kuldeep Kumar, Claudia Martinato, and Sébastien Jacquemont has made a catalog of all of the most common copy number variants in the genome and their effects or their mean effects on the subcortical anatomy using ENIGMA's shape toolbox developed by Boris Gutman. Now, how do you make sense of this? I mean, there's a huge amount of information here. What they then did is they fed all of the common deletions that they had imaging data for into a clustering analysis, found that there were common patterns, or at least patterns that cluster, and that these co-clustered with the idiopathic variations that are seen in schizophrenia and major depression. And I mean, this is a really incredibly interesting way of looking at the link between a deletion in the genome that often confers heightened risk for disease and the pattern that's typically associated with that disease, and seeing whether or not people are more or less likely, based on their imaging, to develop those conditions. So congrats to Kuldeep and Claudia for this great analysis. Our first ENIGMA paper of 2024, congrats to Rune Bøhn and colleagues in Oslo. This idea is to look at the intra-individual variability of the brain. So I mean, obviously, if you carry a CNV, there may be a lot of variability across the brain without there necessarily being a mean difference. I mean, that's a different question. So he and colleagues have been looking at this, looking at intra-individual variation, finding that this is commonly associated with CNVs. If you'd like to take part, new cohorts are always welcome. If you have genome-wide genetics data, they'll help you call CNVs and help you analyze your data there. A few working group updates. Congrats to Odile van den Heuvel and Dan Stein and colleagues in the OCD working group. They've been completing a vast range of projects that I won't read out, but you can look at them here just as a record. Many of them moving from structural and diffusion MRI into functional MRI and looking at a finer scale into the particular structures that we've talked about that you would look at with the parcellation methods and the voxel-wise methods. One interesting result by Nadia Janalia is that they had a tremendous amount of task-based fMRI in OCD, and they found that there was greater task-related activation in patients in regions involved in top-down inhibition. And not only that, they could predict better repetitive TMS response in those with greater planning-related activity on task-based fMRI. So I remember the reviewers of our grant on this said, why are you doing task-based fMRI? And this is a very good reason to do it. You can predict to some degree of accuracy whether these patients will respond to TMS using these features. Congrats to Chris and Ola for their work in bipolar disorder. Again, there's a vast landscape of studies that are currently going on. They met in Barcelona and in other venues. In fact, in San Diego, there at the top right, Pauline Favre from Neurospin is talking about cardiovascular health as a exacerbating factor in bipolar disorder. And on the bottom left, there was a panel that they held on medication effects and BMI effects in bipolar disorder. Very interesting. A big shout out to L'Oreal Dietz and Thomas Hayek. They've been looking in depth at whether obesity in particular and cardiovascular risk in general, for example, training risk scores or plasma lipids, which are treatable to some degree, whether they accentuate the bipolar clinical outcomes and brain deficits, and they find that they are. This is actually a very nice paper that suggests that a lot of the ventricular enlargement that is famous in bipolar is explained by high BMI, which is quite interesting. I mean, if you look at these things as interactive effects, they could be quite informative. Raj Mori and Mark Logue have 41 projects going on using neuroimaging data in PTSD. This one by Jin Yang and Ashley Huggins uses a method that might be interesting to this group. They take voxel-wise surface-based data, do a non-negative matrix factorization, NMF, to come up with clusters or patterns of coherent cortical variation, and find that a number of these are altered in PTSD. It's sort of a data-driven partition of the cortex that may be useful for other disorders. Also, Shizhu and colleagues have been classifying PTSD with a variational autoencoder. Now, you can feed in structural, diffusion, and functional imaging data. It will compress the derived measures using a variational autoencoder into a simple set of features, a bit like PCA, but nonlinear. And then it'll go ahead and do its best to predict PTSD. It can also do biotyping. So this is, if you've processed data from 3 modalities and you want to have a chat with Shizhu about how to use this, it'll also tell you which modalities are best to make a prediction, and within that modality, whether there are any salient features that have done a good job on their own, either for supervised classification or biotyping that might be used to predict outcomes in subgroups of patients. New paper by Yan Kide and the schizotypy group is showing that childhood trauma is an exacerbating factor in schizotypy. Very, very nice work there. Marlena Staginas, who's pictured here, and Yidian Gao, who've been working with Stefan De Brito and Graham Fairchild in the UK, and have assembled the largest neuroimaging study of youths with antisocial behavior and conduct disorder. This has really been a remarkable study. So the overall picture in conduct disorder is one of surface area deficits, which is very typical in neurodevelopmental disorders, but much more profound than many of the coexisting disorders, ADHD, for example. It seems like these effects are rather more severe. This is a study of 10,000 youths. Also a study by Sophie Townend working in this working group, compared in a vast set of data, anxiety disorders, major depression, ADHD, and conduct disorder. And you can see conduct disorder has the greatest effects. And there tends to be some brain signature associated with externalizing or typically externalizing disorders, conduct disorder and ADHD, which is not found in internalizing disorders such as depression and anxiety. And here's Sophie Townend presenting those results the other day. Kudos to your group for doing this in data from 68 sites, which has made a really definitive study. And here's just a handful of studies that are now going on in that group. ENIGMA Epilepsy, which is led by Kerry MacDonald and Sanjay Sisodia, has been looking at a number of things that might be of interest. So Boris Bernhardt's team has developed a set of algorithms to put everything into BIDS format, make all the data ready for a number of workflows, organize it. I mean, they've been centralizing a lot of their at least T1 weighted MRI data, which has enabled them to run AI methods. So artificial intelligent methods such as convolutional neural networks will diagnose a disorder, tell you using salience maps which brain regions were pertinent to the decision. And in their case of epilepsy, it's of interest to classify lateralization of temporal lobe epilepsy. This is work by Eric Kasten and colleagues. And so this would be a lot of fun to try. If your groups are centralizing data, they will advise you on how to run CNNs for different purposes. And they have a number of active studies that you'll see there. And they also met in Dublin, very nice place to have a meeting. The Parkinson's group, you'll see on the right an overview of the sorts of things they've been doing, from tracking cortical and subcortical atrophy to this wave of deficits spreading in the cerebellum to microstructure. Max Lancer has an overview paper on that. We talked about Sukli Liu's stroke group. This paper I wanted to highlight is sort of interesting. So this is work by Emily Olafsson and Amy Kosaiski. And they ask if stroke affects your motor function, but in a clever way. So they overlay the stroke brain lesion maps, which are in bright blue here, on a connectivity database. They say, well, which streamlines tracked using diffusion tractography, which streamlines are interrupted by the stroke, what fraction of them that interconnect a pair of regions are disrupted. And then they project that onto the cortex. So what's happening over on the left there is you'll see the fraction of streamlines disrupted is plotted. And so then you throw away the tractography data and you have a new cortical map. And the cortical map is how much of the innovation of this area was disrupted by the stroke. And then you feed that cortical map into some machine learning. There's a very, very clever sequence of steps. And they have found that this data-driven biomarker does better than the commonly used heuristics in the stroke literature, such as the fraction of the corticospinal tract that has been overlapped by the lesion in predicting motor outcomes in stroke. A sleep group that is led by Masoud Tamasian and Kai Spiegelhalter published a number of really nice papers. Their second paper found no results, also very valid to publish. There is a lack of structural brain alterations associated with insomnia, but I'm being a little bit facetious. They did find some very interesting results in this paper. Antoine Weiss, who's the first author, worked with Hanwen Bai, who also won a Best Poster Prize at the Institute of Medicine of Neuroscience and Medicine in Jülich for this machine learning work. So they fed in their imaging data from people with insomnia, and they asked which method and which set of features predicts insomnia, and they found that there were some that really did do a good job. And they're now pursuing this in terms of understanding its relationship to cognitive activities that someone might do if they were sleep deprived. So congrats to Hanwen for the award in this machine learning work in sleep. The other thing is very interesting. Masoud has developed a webinar program for ENIGMA Sleep, where I think now 9 people have given really superb talks. You can find them at the YouTube channel there on the bottom left, by experts in various domains of sleep and cognitive health and neuroimaging. So you're very much encouraged to have a listen to those. Another method I thought would be fun to outline is the schizophrenia group has been experimenting with 2 different ways of doing disease subtyping. So the first way, and this is a paper led by Narissa Banaj, said in schizophrenia, there are people that have deficit syndrome and there are people that don't have deficit syndrome. So if people are not familiar with this, about a third of patients have enduring negative symptoms, depression, which comes along with a poorer prognosis. And there's a lot of interest in whether that third of patients with so-called deficit schizophrenia have a different structural anatomy of the brain, and they do. So in blue on the left, there are deficits that are severe in people with deficit schizophrenia, whereas the pink and purple areas are showing gray matter thinning in people with non-deficit schizophrenia. So this is one way of subtyping. The other way of subtyping is data-driven. So it's very, very interesting. There's a paper by Yu Xiao Jiang and Jianfeng Feng, who's long been a friend. He used to work in the Imagen project. Sylvain will know Jianfeng very, very well. And he fed schizophrenia regional data into PySustain, which is a staging and subtyping inference algorithm that takes discrete region of interest data. And here's what they found. So they fed in a huge amount of data. This is from all across the world. And they found a frontal first atrophy trajectory in one subtype of schizophrenia. There they are on the top. If you look at the top row, the first areas with atrophy are the frontal cortex. And then there was another subtype that had a more temporally predominant atrophy, but then the atrophy spread forwards into the temporal lobe. Now you could say, well, how on earth is this discovered? The way they do it is they feed all the region of interest data into Sustain, which was developed by Alexandra Young in London, and Leon Axman and their colleagues. It assumes that biomarkers tend to worsen with time or with stage of disease, and worsen means that their Z score or Z score gets worse. And if there's a monotonic trajectory, and that these markers tend to become abnormal in a certain order, maybe frontal before temporal or temporal before frontal, you can have the algorithm sort this all out and give you a sequence. And I mean, this won't always be observed, but it'll work out the most likely ordering of these. And it'll do it for you. It'll assess how many subtypes there are, in this case, two. And they were able to replicate these two subtypes, not just in European ancestry folks, but in people of East Asian ancestry as well. It's very strong evidence of validity for this method. And you can look at these trajectories and see to what degree they're evidenced in East Asia versus other parts of the world. Really interesting subtyping approach. This was a partnership of ENIGMA with the ZIB Consortium, which is a Chinese consortium interested in neuroimaging of schizophrenia. Another approach led by Wolfgang Omlor and colleagues in the Schizophrenia Working Group observed greater variability in brain metrics in schizophrenia, which is also consistent with the subtypes that we just saw. This paper last month in Nature Neuroscience, Rebecca Nickmeyer and Anne Alex made normative charts for infants. So here are boys and girls, and their brain measures were linked with language and motor function. It's a fascinating paper in Nature Neuroscience last month. Some good news is that children born preterm or with low birth weight, they found that they tended to show catch-up in brain growth with age. I mean, that was known to some degree, but they've charted this using normative charts, so congrats to them. And now they're moving on to resting state fMRI. So Wei Gao, here he is at SOBP, showing that the majority of the resting state networks are established by 9 months of age. And he's very keen to bridge infant resting state network analyses to the older pediatric groups to make normative charts. Obviously, there's some difficulty harmonizing this because if you scan a baby with resting state fMRI, the baby may not be awake. Maybe with an older child, they will be. And some children are scanned while watching a movie, so there's obviously need to harmonize the types of data that are going into these studies. ENIGMA Laterality has published 10 papers to date. Thank you to Clyde Franks, Eileen Luders, Shangzhen Kong, Simon Fisher. A rather nice paper in PNAS on the left, led by Dick Shiven, showed that in schizophrenia, the lateralization of the brain is reduced, which had been hypothesized since Eve Johnston and Tim Crow scanned the first schizophrenia patient with CT in the 1970s. And I remember Tim Crow coming to visit us and badgering us that this was the main thing that we should be looking at. So they did, thank you. Tim Crow will be happy. So it's very interesting. There's also a study by Eileen Luders and Florian Kurt showing that the asymmetry of the brain doesn't just increase as life goes on. So they found in blue areas where brain asymmetry is gradually reducing with development and others where it's increasing. And then Shangzhen Kong and colleagues have been looking at brain asymmetry in bipolar disorder. A very interesting series of papers. Now, they also are interested in trying surface-based asymmetry and even voxel-based asymmetry, which involves registering one hemisphere to the other using a nonlinear flow. And there's a series of methods that they've been working on in their lab, beta testing for rollout to ENIGMA. Cross-modality work. I've talked about each modality one at a time, but there's very interesting work by Justine Hansen and Bratislav Misic in Montreal, where they've been reading in ENIGMA's maps of all the different disorders and the measures of deficits, and seeing if the deficits in different conditions relate to patterns of connectivity, patterns of receptor distribution in the cortex, like where are the dopamine and serotonin and noradrenaline receptors, or even cytoarchitecture. So you can imagine that if a certain sector of the cortex is primary versus association cortex, maybe it's less or more likely to be involved in certain disorders. Now, they had a very nice paper in Nature Communications and a toolkit. So I want to tell you a little bit about this. Sarah Lariviere, working in Boris Bernhardt's lab, developed the so-called ENIGMA toolbox. And many of you have tried this. What it does, among many things, is it reads in cortical maps, and you can relate them to maps of histology, such as the distribution of certain cell types in the cortex, to gene expression, in fact, any molecular map. And it's very easy to use. I even went to a workshop and typed Python code, and if I can do it, then I think anyone can. But there's basically a system to compare your cortical maps with maps from other sources. A couple of other highlights will be done in about 5 minutes. I think about 5 minutes left. Enigma India launched in February of 2023. This is an effort to scan elderly people at NIMHANS in Bangalore. They have been, excuse me, also holding workshops. And so there's one going on now. Well, it's a little late in India. It's going on tomorrow, I suppose. And these studies focus on aging, but also are applicable to neuroimage analysis. And these cohorts will be joining many of the ENIGMA studies that we've talked about. They're also collecting a range of other biofluids and plasma markers to link to the imaging that they're collecting. And this is, I'm sorry, I misspoke. This is the workshop that's going on today, December 18th to the 20th. One major goal of Enigma is to increase the diversity of the cohorts. Now, this is not a new idea, but there was a tremendous workshop led by Raj Mori, who co-chairs the PTSD group, Bisu Gilai, who's a professor at Harvard, and colleagues from different countries in Africa, where they've been collecting cohorts with neuroimaging. And so just to sort of show some of the areas where this is improving, there's an effort in Pakistan with scanning in Islamabad, Peshawar, and Karachi, led by Maheen Adamson. They are starting that project. Neda Jahanshad and Annie Gupta at UCLA, USC and UCLA have a Alzheimer's Association grant to scan people of North African, Middle Eastern ancestry in the US. For many years, Ryota Hashimoto has worked with the adult psychiatric groups to study the deficits in the brain in Japanese people with these same disorders as part of a consortium he leads called Kokoro. And then finally, Chao Gan Yan has a very active collaboration led from Beijing between the DIRECT Consortium, which is a Chinese consortium studying MDD, and Lian Schmal and Dick Veltman's Enigma MDD group. So these are looking forward to cross-cultural comparisons of different brain disorders. A very practical event was led by Carrie Esopenko. She and her team study intimate partner violence and traumatic brain injuries and concussions that result. And she leads a confidential support group from people who are affected by intimate partner violence. I just want to say this has been very widely acclaimed in the media. This is one of the British newspapers, The Guardian, that highlighted her work as part of well, let's get it right. It's part of her Pink Concussions network, which is associated with Enigma IPV, in terms of providing reassurance and practical advice. If you want to give anyone money for Christmas, you can give it to Emily. So she has a global educational network where for $9, apparently you can support the enrollment of a student. And she has 800 students registered from 77 countries in Enigma U. Now, this is absolutely wonderful. I mean, these videos have been contributed by many of you, about 15 minutes long. They're at an advanced high school to introductory undergraduate level, and they cover the whole gamut from neuroscience to psychiatry to clinical medicine, topics related to the brain. And if you want to contribute modules, do reach out to Emily. It's really wonderful, and it's always going on. And there's Emily's flyer if you want to send it to anyone, and there's a short video if you'd like them to see it. And also congrats to Emily Dennis for her Early Career Investigator Award at the IBIA in Dublin, Ireland from early this year. Now, I said this was the last call of the year, but we'll contact you in the coming days about an effort led by Dr. Rima Kadura Daouk, who's at Duke University, linking the microbiome to the brain. Now, many of you are familiar with this or even leading efforts in this area. She's interested in applying for funding, and she's a very accomplished researcher. She leads the Alzheimer's gut microbiome project, which is very successful. She's interested in helping groups get microbiome assays and stool samples paid for, and then linked to imaging and all the other clinical questions that you have. So we'll follow up with you shortly about this project. Sophia Tomopoulos and colleagues have created a range of brochures. If you have the PDF of these slides, you can click on these different links. If you'd like to make a brochure for patients and one for researchers, give us a shout. I mean, these are really nice manuals, both for new people who might join in or for trainees or for even participants in the research studies to see what sorts of things are being discovered. We're lucky to have a lot of visitors. If you come to the marina, I'll take you to lunch. So we'll probably go to this place by the lab. And thanks to everyone who's lectured here this year. Here's a selection of speakers. Many of them will be very familiar to you who've spoken here in this past year. Thanks for coming to workshops. There was one in San Diego, one in Montreal. Actually, I think we just ate food in Montreal. I don't think we had a workshop. But it was very pleasant to see everyone, and we'll do those again, probably at SOBP in Texas in the coming year, and possibly in Seoul in Korea. These are some folks presenting at the Human Brain Mapping meeting this year. Another probably familiar faces to many people here presenting posters at OHBM. And this is quite remarkable. I see, Andrea, you are on. This is a film that was made by Dr. Andrea Rimia, and the original film is actually much better quality, and so we'll link to it. But this is the ENIGMA gathering in Montreal earlier this year. And my goodness, I mean, Andrea, thanks for making this tremendous video, which actually showcases Montreal rather well. It makes it look like a rather wonderful place to visit. Except now the slides are stuck permanently. Okay. All right. We're just about to wrap up. Thanks for presenting at all these conferences throughout the world. You'll see different gatherings of different working groups that occurred throughout the year. And many people you'll know. A lot of keynote lectures that were given on the topic of ENIGMA in various parts of the world. And do let Sophia know if you're submitting something to a conference. And so, I mean, she'll promote it to other people and make sure that other people know that you're there and come and see you. And do put in the chat any other conferences that you'll be maybe going to in the coming year. We'll have a significant gathering at SOBP in Texas and also at OHBM in Korea. Now, this is also, there was some division of opinion on whether we should show this, but I'm going to show it anyway. So Raj isn't dead. He's retiring, or at least he's not actually retiring from science. He's retiring from chairing the ENIGMA PTSD group. But I want to bring this up at this stage. I mean, Raj, you've done such a tremendous job in co-leading the PTSD group with Mark Logue. And I know Raj will be going on to other things in addition to his current career. But he'll remain a guiding presence, and Jenny Stevens and Delyn Sun will also be taking over the co-chairing of the PTSD group. And they architected this very elaborate plan where everyone would survey different parts of the ongoing activity. Anyway, thanks, Raj, for all your contributions. I know you're not going anywhere, but I just wanted to give people the heads up that there'll be a change of leadership. Big thank you to Dan Van Rooy, who's gone on to new ventures. Kathy Lawrence, who's here at USC, will be taking over as co-chair of ENIGMA Autism with Jan Buitelaar. So thanks, Dan, for all of your great work as part of ENIGMA ASD. I did want to give a shout out to Scott Mackey, who's been a very influential force in ENIGMA addiction. He's moving on to do many new things, but we've greatly valued, Scott, what you've done over the years. ENIGMA addiction, very ably led by Hugh Garavan, Patricia Conrad, and has a vast list of ongoing projects. So I really appreciate what everyone is doing in ENIGMA addiction. Some job posts, go work with Jim Prisciandaro at MUSC, or Ashley Huggins, or Theo Van Erp. And also, if you would like to post a job, we'll tell you about it, or you can put it on the website right there. There's more general information for members. Do join an all members mail server, and we'll invite you to things. There's a alias email. I think that's just an email for Sophia, and also a frequently asked question for people who are joining. Some of these resources you might find useful if you're interesting new members or new trainees into your lab who are maybe not as familiar as we are with these different things. And there's the social media stuff. And I think that's about it. I don't know, Sophia, if we can do this. That's officially over, and I'll shut up. But I think we're going to have a group picture. And so if I stop, I was just going to end before anyone else, just by thanking everyone here for such a tremendous journey. I think each and every one of you have led us on a journey that we never quite anticipated when this began. Enigma was founded in 2009. I remember several of us thinking about this maybe would have been a good idea. And I mean, I don't think we'd ever dreamed this would become quite as exciting as it's been. So thank you to all of you. And if you're listening to a recording of this, thank you to you as well. There's a few people that obviously couldn't join given the timing. And good luck to us as all of you proceed into the new year. Wishing you all the very best for the holiday, hoping you get some mental rest and a break from us. Probably be what most people are wishing for. I don't know if Dr. Selman, you have a message for all of us in Enigma, and I appreciate you taking time to join us.
Thank you very much. I consider today was an excellent presentation. And I see how, really this is United Nations studying the brain. And probably this consortium will unite the brain and make peace in the world, support peace, and we will all have enjoying the life and health. And thank you very much, Paul. You've done absolutely superb job.
Well, thank you, Dr. Zellman. So just as a final remark before we go, and everyone's welcome to say a few words if you want. I mean, the talk is officially over. If anyone was unable to attend this, either they were too busy or it's not a reasonable time of day, Sophia will send you a recording, which you're very welcome to share with them. And again, have a very nice holiday, and hope everyone is safe, and we'll see you next year.
Thank you, Paul. Bye.
Thanks, Paul.
Thank you.
Merry Christmas.
Bye. Happy holidays.
Thank you, everyone.
Thank you, everyone. Thanks, Paul.
Thanks, everyone. Bye.